Okay, can everybody see me over the no, I'm just joking. It's a great pleasure to be here. Thank you to the organizers for inviting me. It's really privileged to speak on this panel, especially given that Dr. Sridharamani was actually my first advisor working in any laboratory during a summer internship at Columbia. And we've also closely followed and been in discussion with the folks at ILSI and the CTM as they've developed and as our company has developed. And so we're very excited for opportunities to work together moving forward in this very exciting space. So I've prepared a little bit of a different theme of presentation. The question is why standardization in methodologies, why it really matters for an organization like ours, a small therapeutics company. So just by way of introduction, Symbiotic Health is a New York City-based startup. Our focus is leveraging insights from microbiome biology and combining them with innovative drug delivery in order to develop early-stage therapeutics and move them towards clinical validation. So why is it that standardization and reproducible methodologies are critical in an effort like this? It actually turns out that they're absolutely central across multiple topics and multiple critical activities. And so I just wanted to talk a little bit about the generalities of that and then give a couple of examples of how the process of standardization and methodologies as applied to analysis, but especially manufacturing and clinical trial design and endpoint choice have played out in the microbiome field recently. So I don't need to really review this for this audience, but of course the microbiome is increasingly recognized to be effectively a separate organ system which plays a critical role in many different aspects of human physiology and pathophysiology. And we also have very clear evidence, particularly within the infectious disease context, that that injury and depletion of the microbiome is that causes significant risk for disease, sometimes life-threatening and very severe disease. We also know that many things that we do in daily life, but especially administration of broad-spectrum antibiotics, and can injure and deplete the microbiome and elicit this risk. And we really see our role as the development of microbiome restorative and microbiome-sparing therapeutics in order to address this critical challenge. And so just a little bit of a note on the manufacturing of microbiome-based products. I think we can break microbiome. Just to clarify, I'm going to be focusing on the gut microbiome, and I'm going to be focusing on therapeutics development. So development of therapeutics that are targeting processes occurring within the gut microbiome and also inspired from the emerging biology of the gut microbiome. So one of the critical challenges that we ran up against as a company has to do with how do you develop a scalable, standardizable platform for manufacturing and delivering a microbiome-based therapeutic to the right site. Now, microbiome-based therapeutic can be a wide category of things, and I'll go through a couple of different categories, but just to illustrate here, the process of taking a small molecule and certain very specific species of bacteria that have been traditionally used commercially, the so-called probiotics. The process of taking those species and turning them into a product that you can hand to a patient in a clinical trial or even sell to them at a GNC, that is a very well-established process involving many mature technologies. When you start to look at environmentally sensitive microbiome bacteria, which are much more difficult to culture, and where we don't have the same, they're not quite as compatible with industrial scale processing, at least the processes that have been developed historically. And then again, when you start looking at all kinds of other therapeutic candidates that are emerging from the microbiome, we particularly focus on recombinant microbiome-derived proteins, of which there's really an extraordinary variety with all kinds of very interesting biological activity. When you start to think about how are you going to turn that into an preferably oral product that you can hand to a patient that they can swallow and that it gets to the right place, it retains its therapeutic activity, and how can you characterize that and do that in a way that's reproducible and scalable, that really became sort of the core challenge that inspired the formation and continued development of our company. So one of the real interesting opportunities we have in the microbiome field is this incredible opportunity for translational development. So with an explosion of discovery work in the microbiome, which I think is going to be greatly abetted by the kinds of approaches that Dr. Pace mentioned in terms of better standardizing the discovery process and the characterization of the metagenome. Moving forward, there's really this incredible plethora of candidate pathways and candidate therapeutic interventions that we can evaluate. These can include specific strains of bacteria that we're administering in order to replace or supplement the endogenous microbiome, which may be disturbed, or alternatively in order to supplement it in a way that affects a pathophysiological process. They can also involve microbial proteins. There's really a number of very interesting programs using host-derived factors or traditionally used medications that happen to have interesting effects on microbial physiology, and using those in order to tune the physiology of the microbiome in interesting ways. So then that needs to go into, of course, the question of drug delivery and drug formulation. How do you turn that into a tangible product that you can evaluate in a clinical trial, as well as preclinical development? At each of these phases of development, standardization is a critical bottleneck factor. So I'll explain why. So we've talked about some of the issues about reproducibility of in the microbiome discovery field in terms of just exactly what's going on, how do you take an accurate snapshot of the physiology, and how do you then relate that to a disease process. This is a really critical issue on the analysis side, and also especially in terms of animal models. In terms of microbial strains, here we're in a little bit of a better situation. We can borrow the techniques and approaches that are very well established in cell banking and mammalian cell banking, as well as microbial cell banking. And we can also look to very interesting historical precedents, such as the use of BCG for the treatment of bladder cancer, for example, or live bacterial products that are used as vaccines. The vaccine field can also be a very interesting source of precedence in this area. But nevertheless, there is an emerging need to understand what are the appropriate standards of quality and standards of reproducibility for cell banking for microbial products. Then moving into microbial proteins, here again, we can learn a lot from the incredibly successful field of using host-derived mammalian proteins and understanding their quality and understanding their activity and what are the standards for that at different stages of clinical development, but there's still a great deal of work to be done. And then I think the last and perhaps most subtle and most interesting, and I think actually in some ways the area where there's the most opportunity to have a broad implications for general medicine is what exactly are the interventions that we're doing all the time doing to the microbiome, and what does that have to do, what does that mean for human health? And in the case of a microbial product, the most important aspect of that is clinical trial endpoints, and their choice, and that has a critical impact. But also, outside of the context of that, how does microbial metabolism and diversity in microbial metabolism affect clinical outcomes, even with interventions that seem to have nothing to do with the microbiome? And I'll give some examples of that. So, for example, it's increasingly clear that the gut microbiome and diversity in the gut microbiome is a critical factor in drug metabolism, toxicity, and efficacy. And so this has been a very active topic of research in Peter Turnbach's lab, and this is a figure from his lab showing, for example, that the dynamics of metabolism of very, very widely used cardiac drug and the implications for blood levels of this cardiac drug are directly related to the presence or absence, which can be variable, of a particular strain of bacteria within the gut. And it is the activity, the metabolic activity of these bacteria, which is actually transitioning these agents from active to inactive forms. So that and this is a drug which has a very, very narrow therapeutic window. So that means that the beginning state of a patient who walks into the clinic, what is the state of their microbiome, will have a significant implication over what is the likelihood that a drug will either control their cardiac problem or potentially kill them. And that's the ways in which we can assess the state of the microbiome in patients that are undergoing routine but also investigational clinical interventions. I think there's really a significant, really lack of standards about how we can go about doing that, but it's increasingly clear that it's very important. We of course also know in the case of infectious diseases that the gut microbiome, the pre-existing state of the gut microbiome is an absolutely critical risk for illness and infection. And the best example of this is probably in bone marrow transplantation patients. So for patients undergoing allogeneic bone marrow transplantation, when they present to the clinic, have about a 1/3 chance of being colonized at the time of presentation with vancomycin-resistant enterococcus. And this is largely due to the fact that these patients are treated with rounds after round of antibiotics, often for the prevention of Clostridium difficile infection, which I'll talk about in a minute. This leads to in a subset of those patients, a massive overgrowth of vancomycin-resistant enterococcus, which, as is illustrated here in work from Eric Paymer's lab, can actually be detected serially in the stool of patients. These are not patients who have an overt infection with vancomycin-resistant enterococcus, but it's there and it's growing. And it's growing because of the interventions that these patients are undergoing, antibiotic administration. And so as illustrated in the red bars, those patients that have this bloom of vancomycin-resistant enterococcus have an incredibly elevated chance of then getting a vancomycin-resistant enterococcus infection somewhere else in their body, not in their gut, but somewhere else in their body. And of those, the mortality is extremely high in patients that undergo that. So this is really, it's a perfect example of an opportunity for precision medicine. The pre-existing state of the microbiome is going to have a huge impact on the outcome of treatment for these patients, even in situations where we're not ordinarily associating with the gut microbiome, such as bone marrow transplant patients. So with that, I'll talk a little bit about our major focus, which is Clostridium difficile diarrhea. Clostridium difficile is the leading hospital-acquired infection within the developed world. It's a leading cause of infectious and debilitating diarrhea in approximately 500,000 US patients each year, with estimated 30,000, between 15 and 30,000 deaths. Clostridium difficile is the canonical disease of the microbiome, as many of you may know. It arises almost invariably as a complication of treatment with broad-spectrum antibiotics. So I'm going to talk a little bit about our initial experience on fecal-derived microbiota transplantation, because I think this represents the most challenging example of a situation where standardization is critical and also exceptionally difficult. So it's been known for many decades that there's a potential utility of transplanting healthy gut-derived bacteria from donor individuals into individuals with recurrent Clostridium difficile. This causes a reset of the gut microbial ecosystem and often leads to remission from recurrent disease. And as I mentioned, this has been known for some time. The interest in the microbiome field was really catalyzed in 2013 by the publication of the first randomized clinical trial of fecal transplantation for the treatment of recurrent Clostridium difficile. The origins of our company, we worked to develop an oral formulation of this methodology, which typically involves invasive infusion procedures, and we developed the first generation of our drug delivery technology, which was a capsule, enteric-targeted, gastric-targeted capsule for the delivery of transplanted bacteria. We published this work in 2015 showing really excellent remission rates in concordance with other published studies using additional modalities for fecal transplantation. So how exactly can fecal-derived microbiota transplantation work as a scalable commercial process? There's a variety of models which have been explored, including the one which is currently being practiced by my co-founder, an infectious disease physician, individual physician in his own clinic, sources donor material, screens it according to certain accepted clinical criteria, but nothing approaching the level of standardization and reproducibility that is typically expected in a pharmaceutical process, for example. Then you have an alternative strategy, which is called a stool bank, where you have a centralized process that applies a somewhat more streamlined and reproducible process to the processing of fecal-derived microbiota, and then can distribute it to various clinics. So these models are all being, and then finally, of course, you have, I think, what the Food and Drug Administration would prefer, which is that somebody come to them with clear, coherent criteria for how to take human fecal material and turn it into something that it can be acceptable as a drug. And again, the notion that a highly heterogeneous material can be a drug, this is not an unprecedented phenomenon. We have intravenous immunoglobulin, for example, which is a very complex material sourced from a wide variety of individuals. So this is not something that is completely out of the question. It just gets down to how exactly do you characterize two different batches of fecal material. What are the specifications? What are the margins? What are the parameters that we're applying? Is it the number of live cells? Is it their diversity? Is it some high-level genetic index of what their composition is? So, in practice, all of these models are being applied in parallel. Really, we have many independent clinics which are performing fecal-derived microbiota transplantation with a very, very wide, we don't even know the level of variation, very wide variation in how the samples are processed, how they're treated, how they're characterized. There is at least one currently operating stool bank, which is shipping approximately 12,000 units of a basically cell-based therapy per month throughout the United States and in fact around the world. This is operating under a relatively ambiguous regulatory status where the FDA has effectively decided to not enforce guidances that it has in place because in the long run, the use of any fecal-derived, of any microbiome bacteria, whether fecal-derived or cultured, will be under the auspices of the Center for Biologics Evaluation and Research. It will be considered a biologic product in the long run. But the FDA has chosen, in this very specific instance for the treatment of recurrent Clostridium difficile, to not enforce their authority in this respect. In parallel, there's a number of different programs that are seeking to meet the standard FDA criteria for a pharmaceutical product in the case of fecal-derived microbiota transplantation. At present, the most advanced is a company called Rebiotics, which is effectively adapting traditional fecal transplantation methodology with some more stringent screening and characterization processes. And they are entering phase 3 early next year, I believe. So Series Therapeutics, which has been kind of the poster child on the commercial side for a microbiome company, and has really enjoyed enormous financial success as well as recognition for the work that they've done. I think it's a really interesting case in point. The company was launched based on the results of a phase 1 study, which was a relatively small 29-person study in which they used a relatively crude product, very similar in many ways to traditional fecal transplantation, and showed a very significant clinical benefit. A couple of weeks ago, they announced negative results from a phase 2 study where they took the basic idea, which is delivery exclusively of fecal-derived spores from spore-forming bacteria, and they refined it. And they turned it into something much more closely approximating a pharmaceutical product. So they went from 60 pills to 3 pills. They went from a very involved patient experience involving a lavage enema to a patient experience that's much more similar to just taking an antibiotic capsule. This study actually was a failure in terms of efficacy, not in terms of safety. The safety is still quite good. But they were unable to recapitulate the exciting results that they and many others had observed with fecal transplantation, leaving really kind of an open question about product reproducibility and scalability. Did they somehow, in turning their product into a more standard and refined product, did they lose something that was present in the original material? And this kind of gets back to the question is that we don't necessarily know what we are optimizing for, particularly in the case of fecal-derived products, which, as I said, I think is the most challenging one. So I'll move on to what we think is a little bit more of a tractable problem, which is a case of a microbiome-derived recombinant protein. This is our major focus. So our lead program, which we call SHP-01. SHP-01 is an engineered version of a lytic enzyme, which is naturally occurring in bacteriophage. Bacteriophages being viruses that infect bacterial cells, in this case, a bacteriophage which specifically infects Clostridium difficile cells. These lysine enzymes have evolved in order to cleave the cell wall in order to release viral particles. In this case, we've taken this lytic enzyme out of its context, expressed it recombinantly, and applied it to the outside of the cell, this being the work of Dr. Vincent Fischetti at Rockefeller University, from whom we've licensed this particular molecule. So fortuitously, these lysine molecules retain the ability to cleave the bacterial cell wall in a remarkably rapid and efficient way, as observed on the right, and our ability to suppress the load of Clostridium difficile cells by 400-fold in a mouse model of severe disease. Interestingly, they exhibit a very low rate of acquired antimicrobial resistance, which is an increasingly important standard for antimicrobials development as well. So I have let me see if these will work. Okay, I'll just move forward. So one of the really important features that we like about SHP-01 and why we think it has a potential to have a very positive impact is the fact that it's an ultra-narrow spectrum antibiotic. So the concept here is that we're specifically going to eradicate Clostridium difficile cells without injuring the healthy gut bacteria. So this is a preliminary experiment that we've done with Dr. Fischetti on this matter. So comparing vancomycin to SHP-01. Vancomycin, of course, being the standard treatment for Clostridium difficile and a broad spectrum, very important antibiotic. Both vancomycin and SHP-01 being active against a wide variety of clinical strains of Clostridium difficile. But vancomycin is also wiping out the vast majority of both unrelated Clostridial bacteria and related Clostridial bacteria and unrelated representative microbiome bacteria, which SHP-01 is active against only a very, very small minority, supporting its ability to be an ultra-narrow spectrum antimicrobial. So we see this possibility as a supplementation therapy in order to accelerate antibiotics, the efficacy of antibiotics against Clostridium difficile. We also see possibly the combination of ultra-narrow spectrum antimicrobials and transplantation of therapeutic bacteria as a potentially interesting strategy. So in terms of standardization and in terms of what the standards are going to be applied, we are very much looking forward to coming up with a more powerful argument for how to quantify microbiome sparingness as a desirable quality for certain kinds of interventions, including narrow spectrum antibiotics, and really kind of what are going to be the standards moving forward for that, because we believe, in fact, that the ability to target a pathology without injuring the healthy microbiome is increasingly going to be a criteria that's applied not just to our product, but to antimicrobials generally, and in fact, to really a wide variety of medical interventions. So I'm just going to talk a little bit about our work on the manufacturing side in terms of coming up with a standard commercially scalable drug delivery formulation. We use a combination of material science technologies in order to retain bacteria or recombinant proteins in a therapeutically active state while targeting their release to the lower GI tract. Just to illustrate some of the challenges that we faced and progress that we've made, we've come up with a microencapsulation based protocol where we create micro droplets of an aqueous matrix environment, which is favorable to the activity of recombinant proteins and also is conducive to the growth and maintenance of even very finicky anaerobic bacteria, illustrated here. And this is adapting from this is adapting standard technologies using emulsion-based procedures to deliver small molecules. But given that our capsules are significantly larger, they can accommodate these novel therapeutic classes, such as microbiome bacteria. So some data on the tunability of our formulation. We really see our role as a company as taking potentially transformative therapeutic modalities which operate, which are very effective at the laboratory bench, and addressing critical drug delivery bottlenecks in order to turn it into something that you can hand to a patient in a clinical trial. And from that point of view, we really focus on tunability, on the ability to adapt this delivery technology to a very wide range of different APIs and also different physiological conditions, as evidenced here. We can slow down or speed up the release of a representative bacterial enzyme within the simulated gastrointestinal tract. And this is the formulation of novel microbiome-based therapeutics for therapeutic intervention is an area of significant interest, of course. I've discussed the strategy of administering spores, for example, which is being pursued not just by Seres, but also by Johnson & Johnson, and also was the subject of a quite successul program from Biopharma, which was quite ahead of its time as well. And we really fall within the category of polymers and material science-based approaches, and we really focus, as I said, on the flexibility of the platform, the ability, as indicated on the y-axis, to tune the release of the API to particular parts of the gastrointestinal tract, as well as make it compatible with a wide range of therapeutic cargo. So, of course, this is something which is which our major focus being FDA-regulated therapeutics, but there are potential applications in consumer-facing wellness products, and as was discussed, the question of alternatives to antibiotics within the agricultural industry is one that is of great concern. So just a little bit of acknowledgments. We've benefited from a lot of great advice from the LC team since for some time as we've developed as a company. I am the founder and CEO. I come from a pathogenesis background. Our CTO, Noel Godard, is here, and she leads up our biomaterials and drug delivery efforts. And the inventor of the lysine, which I mentioned as an example of a non-bacterial microbiome therapeutic, is Dr. Vincent Fischetti at Rockefeller University, and we're funded by Topsmith Partners, a local venture capital fund. So I hope I've been able to convey the importance of standardization and reproducibility in the context of an early stage therapeutics program, and not stretch the theme too much, but I would very much welcome any questions you may have, and thank you for your kind attention.
So one of your competitors, Actogenics, is using lactococcus to deliver therapeutics to the gut. It wasn't clear, are you taking a similar approach?
We're not, actually, so we deliberately shy away from any administration of any genetically modified organisms. So Actogenics, as I understand it, was recently purchased by Intrexon and was kind of folded into their synthetic biology program. There are a number of other synthetic biology-oriented companies. Synlogic is another one that's received a great deal of attention, that's working with some technology out of NYU for basically, and just to introduce other individuals in the audience that aren't as familiar, there is a great deal of interest in taking organisms which have this exceptional safety profile, such as the commercially available probiotics, and tailoring them in some fashion to use them to correct metabolic pathways which may be missing, for example, in rare genetic diseases, such as diseases of phenylalanine metabolism, and separately to replace metabolites that might be absent in certain individuals. The other major application, of course, is vaccine delivery, which is an area of significant interest, particularly using spores. We're specifically not doing that. is a pretty significant track record of the FDA approving unmodified, naturally occurring organisms for administration to patients, like, for example, BCG for bladder cancer or the oral typhoid vaccine. Once you start to talk about genetically modified organisms, a lot of questions arise about the questions like transmissibility of those genetic elements, and I think that was a level of regulatory risk that we were not particularly interested in overcoming, aside from the significant technical challenges of doing genetics in non-traditional genetic model organisms.
Also, I think it points out with least actogenics the problems of using mice, because they had put IL-10 into the lactococcus and found it was really effective against IBD in mice. And then they went to human clinical trials, totally failed. And it turns out that the bile acids in human were killing the lactococcus, but in mice they don't have the same bile acid profiles, and so they were not getting killed. So it set them back considerably, and they had to generate a lactococcus that was resistant to bile acids. So that's one of the problems, as you pointed out. So any other questions here?
So how frequently do you have to administer the lytic enzyme?
We are in the in vivo data that we have so far, which is from a mouse model. A single dose administered by enema does, in fact, provides some protection, not complete protection from mortality. Moving forward, we are looking at a once daily administration for 5 days. For our second species.
I kind of had a related question to that. So you're relying on enteric-coated capsules to deliver this, right?
Okay, so we use a combination of different technologies for that. So first is we do have enteric-coated capsules that will basically deliver into the small intestine, protect the protein from the acid. But then there's a secondary question: is we ideally want the payload to be delivered to the lower gastrointestinal tract, right? The most targeting technologies are focused on getting things into the small intestine so that they can be absorbed, whereas we have a very different kind of challenge. We want to protect the therapeutic protein from digestion in the small intestine. So for that, we have a variety of technologies that we've developed in order to address that question. But effectively, it's a stabilized macroemulsion whose breakdown is triggered by time and also by the presence of certain ionic factors that are present within the GI system. And the idea being that we can delay the exposure of the API to digestive enzymes until such a time as they are at the site of pathology within the lower GI tract and can be released.
I guess my question is, so you require the patient to take the capsules in fasted state every day then?
So we are, as I mentioned, we are preclinical company, but these kinds of considerations of how the GI environment, physiology of the GI environment is going to vary between different patients are really critical. No, the answer is no, and the reason is that some of the triggers that we are using in order to open the capsule and release the content are in fact of a dietary origin. So we don't anticipate requiring, and we certainly in our animal experiments are not designing concurrent dietary manipulations beyond normal feeding. Yeah.
Other questions? If not, I'll say thank you. And we're going to take a break now, Joanne. Time for a break? 15 minutes?